I'm going to start with the acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australia and Australian and global society. So I am really happy to have Cheng Chen here. So I've known of Chen for a long time and known his supervisors for a really long time. So I think other people in the room will as well. So he's from Xishuangbanna, which if people don't know, it's one of the coolest rainforests in the world because it's tucked up really high in interior southern China and gets the monsoon fog that keeps it a rainforest, even though it's normally out of what would be a rainforest area. Tons of beautiful wildlife. So it's no surprise that after doing his undergraduate in biotechnology, he switched to ecology. And then he went to the University of British Columbia and was a master's student with Jed Brody, who gave a talk here at CBCS about a year ago. And then after doing his master's at UBC, he stayed there and became a PhD student with Cole Burton, who is a longtime friend of myself and also has worked in and around Australia from time to time. So Cheng is going to tell us about a few different projects, and that might take a while. So I'm going to let him take over now and hope that there's time at the end for us all to ask questions. And he's here luckily because he's celebrating his wife's graduation, delayed due to COVID. She went to the University of Sydney, so he's up here just for one day. But if anyone would like to get in contact with him later, just ask me and I'll give you his WhatsApp. You can call him anytime. Just kidding. No.
All right.
Thank you, Cheng.
Yeah. Matthew for the introduction, and I'm really honored to be here, and thanks for having me and coming to the talk. So today I'm gonna talk about the mammal community response to human disturbance and the insights from global camera trap surveys. As Matthew introduced me, so this is a picture of a group of elephants leaving the Xishuangbanna and migrating to the north to find their new home, and they eventually come back. But this is a great picture to capture this how mammal respond to human disturbance. And as Matt has mentioned before, I'm from this region. It's called Xishuangbanna, which shared border with Myanmar and Laos, and as Matt mentioned, it's the northern range of tropical rainforest, and it has a unique landscape as well as the ethnicity of different groups of an ethnicity group of people living there. So before I went into my specific research, I want to just give you a bit of background about the ideas that brought me to this this research career. So I grew up in Xishuangbanna, and before I moved to North America to do my master's, I was a field assistant who helps a professor install the camera traps in this strictly protected national parks in Xishuangbanna. And this is one day at what we find. It's basically a group of poachers or hunters. They killed a bunch of birds. You can see there's birds over there, and they also built this sort of picnic table from bamboo within like strictly managed and protected area, and had their barbecue birds. So that got me. So this is pretty much the first time that I have encountered this issue, which I will talk about in a minute. At the sort of face-to-face experience to this issue, which is the Anthropocene defaunation. So Anthropocene defaunation is a sort of special, it's a special sort of issue of the ongoing sixth mass extinction. And according to the IUCN, if we look at a global scale, 27% of mammal and 13% of birds are endangered. And to So to start this alarming trend, we need data, and we need to know where the species is distributed, how abundant they are, and in the specific areas, and how they respond to exponential disturbance. Of course, it's difficult to conduct research on every species for all and to know all together all the information for them. So people often rely on this high level biodiversity indicators such as the IUCN Relativus, and which I mentioned before, and also the Living Planet Index. Living Planet Index, which is just a database that people gather data from all the abundance data that they have to make this prediction for the future population change. And there's some issue with this kind of high level biodiversity indicators, which they are often known as retrospective late warning signals, which means when you see the change in these index, the change already happened, and there's like a time inherited in these indicators. Therefore, so the GeoBomb have come up with this idea, which is called essential biodiversity variables. The point of this is you need to monitor some of the essential biodiversity variables at the basic fundamental level, and to monitor the change of these variables, and these variables can detect the early signal of biodiversity change, and eventually come back to the higher, to feed the information back to the higher level indicators, and these variables can be monitored globally. So, so the recent remote cameras, or known as camera traps, enables us researchers to better monitor the changing abundance and the distribution and mammal communities, especially for large to mid-sized mammal. And camera trap studies are going, the number of studies that use camera traps are going exponentially over the past 2 decades. And the camera traps are a great tool to estimate the essential biodiversity variables especially for mammals. But there are few studies have pulled data together to look at conservation or ecological questions. And few examples included this one, which they assessed the global pattern of mammal communal occupancy, and of course this one from Matthew's study, where they look at the megafauna extinctions across a large scale, a regional scale. So for my research, I have this core question, which is I'm trying to understand how diversity of large to mid-sized mammals are associated with human or anthropogenic disturbances. To do that, so camera traps is a unique tool, and we'll start from the easiest one. If you have a camera trap and you capture a species, then you have a solid distribution data. You know where it's distributed. And if you count how many species you detected from the camera, you have this diversity data. And if you count how many times one species is detected over a amount of time, you have this abundance data. So my research are focusing on this 3 sort of biodiversity metrics, and I will present 4 related research on these 3 metrics. So that's the 4 research that I'm gonna cover today. And before I get into the specific researchers, I'll just talk about a little bit of the data I'm using. So I started with the team and eMammal. All of these are the camera trap data sharing platform, so people can upload their camera trap data into the platform. Of course, I've gathered data from all the colleagues, my lab mates, my old data and friends. Also, from published studies. The team and the e-mammals now have been built into this big data platform called Wildlife Insights. So I've got about 9,000 camera traps across 28 countries in total. One thing I do want to mention is that I only included the projects that have more than 400 camera trap days as a minimal sampling effort. That's not a lot, but only a few projects are close to this number, but the average mean camera trap days of the projects that I'm using are around 5,000. So I'm going to talk about the distribution a little bit. In this study, I compared the occurrence data that generated by the camera traps with the IUCN range map and see how they differ from each other. So if you want to know what species distributed in a given area, the go-to sort of source is this IUCN range map. We started with people just drawing the polygon for the known distribution limits for one species. It's pretty straightforward. We can just look at the map, and the map is pretty coarser, and it doesn't include all of the details for the distribution of that species. So that brings two potential issues of the IUCN range map. The first one is the accuracy. So sometimes it has been recognized to underestimate the complete geographic range of the rare species and overestimate the geographic range of common species. And people have been criticized this. It's infrequently updated. Of course, it depends on which taxonomy you're looking at. But for mammals, it typically get updated every 5 to 20 years. And of course, accuracy for mammal range marks are less validated by our occurrence data, simply because we just don't have that occurrence data available out there. And the most importantly is that the way that people use IUCN range map, they treated it as an accuracy distribution map. They sort of do this kind of thing, like they overlay all the range maps they can find in a given location and generate this kind of a richness map or hotspots and identify the hotspots to guide the conservation actions. So so in this chapter, in this research, I want to know what is the level of agreement between IUCN range map and occurrence derived from camera traps for the mammals, and what factors affect the level of such level of agreement between these two sources with the occurrence data. So to walk you through a little bit how this modeling for the method work. So if you have, so let's say the green square is a camera grid. You have a camera trap survey, and the blue polygon is your IUCN range map. So I'll start with the B scenario. If you have a camera trap survey within the polygon of a species range, and then you detect that species. That's the first sort of perfect scenario. You have a IUCN that is here, and we detect that. But sometimes it gets a little bit tricky. For example, you have your camera trap survey, but you didn't get that species that's supposed to be here according to IUCN. So you have this A scenario, and sometimes you get you have a camera trap survey outside that species range, but you get that species. So that's the three scenarios that you can would encounter. So I'll give you a little bit more to help you to visualize how that works. I'll give you a real example. So this is a marble cat. For example, in some Borneo places, we set up a camera and we detect that cat, but the IUCN range map didn't include this area. But for some projects in Thailand, we know the IUCN says, oh, the cat's in this park, but we didn't get one by the camera. All right. So I sort of summarize all the species times size occurrence or detections from the data sets I just presented, and I got almost like 3,000 sort of pairs of species times sites. I found like around 40% of them are following this both scenario. IUCN only are fixed about 60%, and only 4% of them are camera only. So to make this further analysis more feasible, I sort of recoded them into I coded the both into the 1 and the other 2 scenario into the 0. So in which case I can run 2 separate binomial regressions models by extracting the data from this part. For example, I use both and camera only data to run this binomial model to see which factor will contribute to the probabilities of a species falling into the specific category. So, so this is our results, and to help you better understand that, I sort of changed the y-axis into the probability of getting the both scenario. So as you can see, the body size matters. If you have a larger species, you're more likely to get that species detected by the camera. Also, within the IUCN range. Also, the species generalist also seems like to be following this direction, which means if you are a species, which means if you are a generalist, you are more likely to fall into this both scenario. And this is the result for the IUCN only model, which means it's the right part. I took the data from both and IUCN and run the model, and I got this. And of course, it's related to the sampling efforts, which means you have more camera trap sampling, and the more likely you are getting that species. And there are no no significant relationships for the camera only and both model, simply because there are only a few species and sites are camera only, so they're just not enough for short power of the model. Yeah, so I'll sum up a little bit the findings that I found from this research. So few species are detected by the camera outside the IUCN range map, which means IUCN did a pretty good to summarize the sort of the distribution limits of the species, and the camera traps detect less than half of the species that were expected to occur in the survey areas based on IUCN range. And the discrepancy between these two sources are associated with body size of the species, and then the habitat, whether they are generalists or specialists, the habitat groups of that species. And such discrepancy could be contributed by the partially due to the range overestimation of the range map itself, and also imperfect detection of the camera trap surveys. All right, so let's move on to the next part of this presentation, which I talk about a little bit about the factors affecting the diversity of mammal. All right, so you should be pretty familiar with this map, which is the human footprint index. As you can see, it shows that humans have turned large-scale landscape into city, cropland, and roads, and which caused the declines of biodiversity. And setting up and maintaining a habitat, protected area is one of the most fundamental conservation actions that we will do to stop this biodiversity loss. But the effectiveness of protected area has been questioned for a very long time. In fact, this paper showed that even that within a protected area, 1/3 of the global protected land is under intense human pressure. So, so the aim for this research is to evaluate the relationship between the mammal diversity and 3 key indicators of anthropogenic pressures. So I used human footprint, which I showed you before, and human accessibility, which is a measurement of the time that needed from any given location to the nearest town, and the protected area coverage. And in this research, I I not only look at the taxonomy diversity, but I also look at the functional diversity. So so the functional diversity, I took this picture that actually made for our paper, that even though you're getting the same number of taxonomy diversity for 2 communities, but there one could be have low ecological functions for that community because they're in a similar the species within that community are much more similar, like functionally. And I have to because all the camera traps that studies that the data from all the camera trap studies that I use are from for different purposes and they're designed in different ways. So I have to use this metric, which I call that the protected area coverage, which basically calculates how much of the camera trap sampled area overlap with any kind of protected area in the world protected area database. As you can see, some of them are completely outside the park, some are like in between, some are this one is like that we're mostly familiar with is sampling mostly within the park. So you can think of this metric as a measurement of that captures the sort of level of protection in the camera trap sampled area. All right, so that's the main results. It's a lot of information, but no worries, I'll walk you through this. So let's look at the taxonomy diversity in the corner. So what I find is taxonomy diversity is positively correlated with the protected area globally. So the little red one, the little red bar, and the protected area coverage are also associated with taxonomy diversity of species in the Indomalaya region. But it's not the taxonomy diversity of mammal is not associated with human footprint, nor the human accessibility. So it makes like a protected area one of the most important metric, one of the most important human anthropogenic covariate for the taxonomy diversity of mammal globally. The functional diversity only positively correlated with protected area in the near Arctic and North America. Oops, I don't know why it feels this. Okay. So anyway, this one, if you can grab the ground. So on the x-axis, I have 100% protected area coverage to 0% protected area coverage, and the brown red dot is the taxonomy diversity, and the green one is the functional diversity. So this graph is to show that the the taxonomy diversity drops in a faster rate than the functional diversity with the decrease of the protected area coverage. I have 2 potential explanations for why this is happening. One could be like species-rich mammal communities are also that associated with parks are also tends to be functionally redundant. That's one reason. Another reason is like a few species lost within protected area that are functionally unique, which maintain a high taxonomy diversity as well, has also been lost by the parks. Yeah. So to sum up this research, I found that taxonomy diversity of mammal is positively associated with protected area at a global scale, and the association is such an association is stronger than those between the diversity and the environmental factors or the other human disturbance metrics such as human footprint and accessibility. And taxonomy diversity was more strongly related to the protected area coverage than functional diversity. And camera traps, of course, can facilitate such rapid assessment of global mammal diversity and ultimately support more efficient conservation. All right. So speaking of the protected area, we know that it's even though we have like global sort of scale looks at this problem, but we know that there's lots of nuance of whether a park can really work. For example, people are saying that you need to local support. You need to have your local villagers who help you to against the poachers from outside. Then this part can work. Or people are saying like the positive contribution. This is the meta-analysis that people saying like positive contribution to the effectiveness of protected area. Everybody reports the anti-poaching actions is really important for a effective park. So I did a little research using the camera traps, and I evaluated the relationship between the mammal diversity and the prey abundance with the law enforcement and the community outreach of the park. And I go to the 6 parks in Xishuangbanna, and I did a questionnaire survey and asked the people, the local people, like how often do you recognize like the community outreach activities happens in your village? Like how often like the park staff come to your village and do that educational or outreach activities, and how many people got punished by the parks in your village? That asked them the kind of question, and I linked that sort of outreach and the enforcement frequency with the mammal diversity that I calculated based on a camera trap I set inside the park. And also, interestingly, I also got the park reported the number for this frequency. And this is the region that I did. So I surveyed a bunch of the villages outside the park, just right next to the park, and I did camera traps inside the park. And what I find is really interesting is that the size of the park and the village reported outreach frequency are associated with the richness of the park, of the mammal inside the park, and the punishment. The village reported punishment right here are associated with the abundance of 2 commonly hunted species: the white bull, white pig, and the white moonjack, but not with the smaller ones like civet, like 2 civets. Yeah, and all of them are only associated with the villager reported outreach and punishment frequency, but not with the park reported frequency. So I found that pretty interesting. Yeah. All right, so let's move on to I think that would be the board's research. Sorry, this is the summary of the third research today. So local people's perception of law enforcement was positively related to the local abundance of 2 large hunted species, but not related to the abundance of small mammals, nor to the park reported level of enforcement. And the local people's perception of outreach was significantly and positively related to the mammal species richness. And I had this sort of the first and the last bit of this link, which is you have a conservation action and they have a conservation outcome. But this research kind of lack of the two key components of this chain, which you have the attitude change and behavioral change, and eventually get you the conservation outcome. I hope the future research can fill in this gap. Yeah, so that's this one. Lastly, I'll talk about the abundance. So this paper is in preparation, and I want to know the winners or losers in Anthropocene. And then I want to know, do species traits predict respond to anthropogenic disturbance? So we know that this paper, we know that species don't respond to the disturbance equally. Some species handles very well. Some species handles very poorly. And for example, this paper, they think that that there are some traits promoting that's the species' ability of living with humans. For example, back at that time, we call that our selected traits like small size and high fecundity. For example, whether they are widespread, generalist, they are more likely to handling the disturbance well. And yeah, species respond to environmental changes often depends on their ecological and life history traits. And it's more than extinction, it's more about the population in this program, because you're more likely to see the decline of a population for quite a while than you see the extinction. So that's why in this chapter I want to focus on abundance a little bit more, to see that the change, the species response in their abundance to the human disturbance. So I ask three main questions. First, do species traits predict mammal respond to anthropogenic disturbance, and whether functional groups of mammals show differential response to relative abundance to human disturbance. And I use two metrics. The direct sort of proxy of the direct disturbance is the distance to human settlement to the villages, and another human impacts on the habitat metrics. I use this one, it's called the edge influence, which accounts the distance from a given location to the nearest forest edge, and the metric, like the configuration of the forest at that given location. So you calculate how much of a given sampled location influenced by the forest edge. For this research, I used a subset of data from my big data set, which comes from 6 South American projects. These are the map for this 6 projects. They're all part of the team network, and they have a very standardized design for all of it. And then they sample this location every year for at least 30 days, and then sample this place for multiple years. And before I get into to look at how species responded to the human disturbance, I group the 30 species that detected within this in this region by using 9 species traits that I thought are associated with ecological function and the ability of species are responding to the human disturbance. For example, whether they're of course we have body mass, which is the basic, and then the habitat breadth, which captures whether it's a generalist or a specialist. And I have a social, which means whether the species is moving in a group or they're solitary, how the lifespan, and whether they're nocturnal, their diet, and then their litter size, and then their brain size, and then whether they have ability to climb. And I've got 4 clusters, which is the nocturnal, solitary, and opossum, because they're quite unique in terms of the litter size, and then scansorial, carnivores, which is basically like climbing, the species that within the carnivorous order that have ability to climb. And also the last one is the large-bodied terrestrial species. I'll give you a little bit more detail on this. So basically, this is so for the nocturnal salamandra, we have Tomodon and then one, and this is the rabbits and then tapirs and the two tapirs and one small deer. And for the largest terrestrial species, we have a white-tailed deer and peccary and two other kind of deer species. And then for those scansorial carnivores, we have like leopard, puma, and panthera, and then all the way to the South American raccoon and opossums, they're just with them by themselves. All right, I now use this multi-species mixed model to model the how species respond to human disturbance in terms of their abundance. This model can allow me to model the mean group response and obtain a better estimation for whale species by sort of borrowing the strands from the species with similar trait. And I can include this year-side random effect in the model, and it, of course, comes with detection probability. And here's what I got so far. So I find that a positive correlation between the large-bodied terrestrial species with the distance to human settlements, which means that the further they're away from the human settlements, they're more abundant. I also find that there's a negative correlation between the climbing carnivores, meaning that they're kind of okay living close to the human settlements. But I find no significant pattern for the edge influence in this study. All right, so I'll sum up a little bit. That's a lot of information, but so large-bodied terrestrial species are potential losers. The scansorial, the climbing carnivores are potential winners. And the effects of forest fragmentation of species abundance remain inconclusive. And the multiple trait-based clustering provided more insights than just look at the species by using the single species approach. And we know that we are now entering the post-2020 sort of biodiversity era. So we now have the this Kunming and Montreal biodiversity framework, and we have some really ambitious goal. For example, like halting all the human-induced extinction of all known threatened species, halted by 2050, and some other even more ambitious goal. For example, 30 by 30, we want to protect all the 30% land by the 2030, which is only 7 years from now. All of this need robust monitor program. And I want to end my talk here by talking about this paper. He brought up this idea of the fully automated monitor of ecological communities. What they're describing is basically you have your first step, which is you have your data collection, whether you have a camera trap image or acoustic data or eDNA, all of them goes into this data pipeline through the storage and transfer and then knowledge extraction, and then you get all the traits data, abundance, distribution, interaction data out of the sort of data pipeline. Of course, I hope my sort of research provided sort of a good example of a tiny little prototype product of how large-scale monitoring program can be used to answering such ecological and conservation questions at different spatial scale. And I want to summarize that that protected area and the human disturbance are associated with mammal distribution, diversity, abundance at different spatial scale. Camera traps is a great tool to help detect the bias of the range map and improve our understanding of species distribution. And you can generate a standardized data, and it's a effective tool to assessing the response of mammal communities to anthropogenic disturbance at a global, regional, and a local scale. With that, I would like to thank all of the people that helped me over the past 10 years, and all the co-authors and the field assistants and volunteers who helped me clean the data, and then my supervisor, Jonathan Jett, for the guidance. And thank you all for listening.
Thank you, Cheng. Should we have any questions?
I have a question about your the global comparison of camera traps within protected areas and outside of protected areas. So I'm wondering how, if you had to address that, I assume in some spots you've got lots of PA data, but not kind of equivalent outside of protected area data.
Yeah.
And even, I think you also showed an example of when there's camera trap surveys that don't really span the border of a protected area.
Yeah.
It's just kind of all inside or all outside.
Yeah.
You know, how did you account for not having equivalency like that?
It's I have like a background slides in here to show the. Can I just?
It's not running.
Oh, it's not.
It's running.
Can I just click more? So yeah, it's obviously, yeah, we acknowledge that there's like a sampling bias because that's the data we got. Oh, okay. Great. So yeah, so let's look at the first one, the protected area. Obviously, in the neotropic, most of them are within like 100% within the area, but for the Indomalaya, we got a little bit both on the two sides, and then for the near Arctic, we have like North America, most of them are outside the parks, and then most of the Africa data that we use are within the parks. So that what I'm just trying to be like transparent. Here's the data, the histogram that I have. I think we have if we sum them together to run a global analysis, I think we have a gradient for a little bit both. But if you look at the specific regions, that could be an issue for some of the regions. But we just acknowledge this and then be transparent about how the data looks like and show them. Yeah.
You're missing a continent.
I am.
Where's Australia?
Yeah. Yeah, that's because I don't know you back in.
There's just a little long. There's no data over here yet. We're working on it, but.
Two questions. One's related to that question. I mean, how many of these people putting out camera trap grids seem to have a reasonable experimental design? So, what question are they asking, or are they just thinking, "I'm just going to get a lot of camera traps"?
Okay.
Without, I mean, or are they asking questions not about these things?
Yeah. So I have some criteria for including the data that I'm going to use. First of all, you have to focus on not just one species. You have to give me all the data, all the species, the data for all the species that you detected. And then second of all, you can't use bait because that's gonna affect the detection probability. Translation. Yeah. So third, you need to have enough sampling effort in a given area. So I don't have like specific standard for like how many cameras you have to use, but it's just like if you have a few cameras, you just need to sample a long enough time. If you have more camera, you can sample like a shorter amount of time. But you need to have that, hit that minimum 400 camera trap days. Yeah.
I'm sort of really interested. What are their questions?
Oh,
What they're clearly not asking questions about
no.
protected areas.
Yeah.
Because then their design is terrible.
They're all over
Everything.
everything. Pretty much. Yeah. Some is focused on the they just want to compare the diversity inside and outside the park. Some are just looking at whether they can find some bears, for example, in North America. Yeah, all kinds of.
Yeah, so the second quick question, so the human footprint index has a component of human accessibility in it, as you know. Because the human footprint index is made up of what, 8 things.
Yeah.
So they're sort of confounded. Were you able to, or had you thought of just pulling the human footprint index apart, because it's made up of multiple metrics, and putting them all in separately, as opposed to putting them in as an index?
I haven't thought about that, but for the first part, I run a correlation between these two metrics. So they're not strong, at least not the R, there's an R is not about like 0.6. So they're not these two metrics are
Because human accessibility is one of the 8 things in there.
okay.
Yes. So that's human footprint is made up of 8 things.
Yeah,
At least.
yeah,
And one of them is
okay,
accessibility.
yeah.
Distance to roads.
Right. But yeah, but it's a good point. I haven't done that.
I think that would be informative, just pulling that human footprint,
Okay.
because people use these indices like that and the human modification index, but in fact they're composite indices.
Right, yeah, you're right. It's kind of hard to interpret and then like what actually contributed, because they are like a complicated index. Yeah.
If you get.
Yeah.
I'll ask a question then. Thanks for your presentation. Am I right in thinking that one of your conclusions from like your second study was that in protected areas there are higher biodiversity metrics for mammals, right? And then on the basis of that, like if there's also probably like a bias around about where protected areas set up, like probably chosen based on like high mammal diversity, particularly when looking at large mammals, yes. So I wonder like what I didn't quite understand or get the way you set up your analysis to identify those correlations. If it's just in sort of like spatial extent, then I guess then you can't really pick apart that bias.
But yeah, so because of the time constraint, I couldn't present all the details, but this is the table of the variables that I used in that specific research. I included not all, but most of the environmental variables that I think are contributed to the diversity of that area. For example, the temperature, the elevation, and I included like the geographic realm of that area and habitat structure. I have a DHI, which is basically the DVI, the vegetation productivity, and also the canopy height. So I have all this variables that I have in my model. So all the models have this part. So it's like kind of set up as a control. And then I have the null model, or I can't call it a null model because it has the environmental variables in that model. So I call that core model. So the core model is just the environmental model. And then I have the model of my like testing model that have different combination of the human, the 3 matrix that I have, and I compare the AIC of these models. And all of the model has the human disturbance sort of covariates outperformed the core model. Yeah, so that's how I control the difference from whether that place originally have higher diversity.
Just wondering as well, because in with those protected area data, I guess you have like a date when it was introduced as well. And so do you have any like time series component to your data that you then used to be like before, after?
No, I couldn't do that. I think that's one of the points that haven't been like solved. But so basically all the camera trap data that I have are most of them are 2010.
Yeah.
2010 onwards. So I'm kind of just using as this like a snapshot of this 10 years or 20 years. Yeah,
That's great.
yeah.
Thanks very much.
That's a good question. I don't think anyone's looked at time since protection.
I mean, but most of those gazetted areas be quite old compared to like marine protected area, for example.
And like also something getting gazetted and then actually being implemented with management those, that you might need to add in 5 years, you know, or make some kind of window when you actually count it as being effectively protected, if at all, you know, I mean.
We did, done it for birds, the woman who did her master's here. And they had before and after protection and then times of protection. Because people have been collecting data on birds for like a long time. The cameras are so recent that most of the protected areas are already available around when it started. But it's an issue that hasn't been addressed fully yet and is a good topic for someone's PhD in the future.
Agree.
Okay, well, any other questions? Let's thank Cheng, thank you very much.